team. All right, team. Let's hope that my cat doesn't lose his mind here in the background. So as we start doing this, this is about a refresh on the cell cycle from class, and then I'm gonna set up the model for you. This model worksheet that you have, I'm gonna set it up for you so that you find it more doable and you don't feel like you're completely like just off wandering. Normally this is an activity that we would have started in class and started with a discussion and yada yada. And so the fact that I'm kind of leaving you on your own to do it, I wanted to give you a little bit of a setup for it. So for starters, let's go ahead and let's take a look, excuse me, at the cell cycle here. And just wanted to highlight a couple of things, right? So for starters, if we start up here, we're going to go through G1. I really wanted to clarify that everything goes through G1. So all cells are going to expand enough that they become essentially like full, content, happy, like proper sized cells. So your cells have a range, just like you do, of size that they can be. And so G1, all cells, predominantly all cells are going to go through it. In that stage, you're essentially just making more organelles and you're making more of the structures within the cell. At that point, you're going to go through that really important checkpoint. And that checkpoint is, are you healthy? Are you ready and prepared to undergo mitosis? And do we need you to undergo mitosis? And if the answer is no, which it almost always is, you go to G0. And essentially it's a sign that says do not proceed. It doesn't mean you're faulty necessarily. It just means that you are not going to undergo mitosis, or at least you're not going to undergo mitosis at this moment. And essentially what happens is all your cells in G0 are doing their normal cell jobs. And I wanted to really highlight this concept that you're either doing your normal cell job or you're doing mitosis. You don't get to do both. And that's why it's very important. And that's why we have so many cells in this G0 stage, because we need the majority of our cells not to be growing. Quite honestly, we need the majority of our cells to be functioning and doing the things that they need to do to accomplish, the tasks of life. So then from there, let's assume you do go through and they've essentially said, yep, you can go through. Now we're going to go into replicating DNA. This is the S phase. S is for synthesis, synthesis of DNA, replicate DNA. Sometimes I say duplicate DNA. All of that is really, really important. And this is where, in terms of an image I can give you, we go from 100% of our DNA to 200% of our DNA. And so often when we look at chromosomes, we think about chromosomes as an X. But what's really interesting and not necessarily intuitive right off the bat is that actually a chromosome looks more like this. It's one long stringy line. And so what happens when we duplicate it, that's when you get the X. So that's when you have one of those chromosomes there, one of those chromosomes there. They're connected by this little thing called a centromere. That's what the spindle fibers will actually attach to. But when you think about the X, all nice and condensed, first of all, it is not condensed at this point. So it wouldn't look like an X. Instead, it would look like, so if you had your one long string, then you would have kind of your second long string, and they would still be connected by a centromere, but they wouldn't be nice and condensed at this point. But this is where you get that X looking thing from. That's actually two sister chromatids. So at this point, you have sister 1 and you have sister 2. These are absolutely identical copies of DNA. So we go through we go through synthesis and we make it back to growth 2, and growth 2 is essentially preparing to become two cells. Do I have enough endoplasmic, Golgi, ribosomes? Do I have enough mitochondria? Do I have enough of all the things that I need in order to inevitably split into two cells that are both functional? So that's really what it's checking. And then once you get there, now you've actually gotten into this area of mitosis. And this is, so you have to do all these other things just before you even get to mitosis. And so in the stages of mitosis, right, we talked about prophase, which was losing the nucleus and condensing, then metaphase, lining up in the middle, all right, so that we can break apart the chromosomes, those sister chromosomes, and put one into each side of the cell. Anaphase, the actual pulling apart of the chromosomes, and then telophase, the decondensing, the reformation of a nucleus, and then ultimately there's this other part in here called cytokinesis, which is actually the pinching off. It kind of looks like this, and then it will pinch off until you become two distinct separated cells. So all that is kind of involved in the cell process in that capacity. We can kind of say cytokinesis is right here. Like we can give it its own little place. There's cytokinesis. I just drew it in. Obviously, it's not before start, so we have to kind of move start. So then start would be right here, and cytokinesis is that black shaded in part. So this is essentially the cell cycle in a nutshell. Hopefully that helped out. Now from there, we're going to move on to this conversation about mitosis and this other process of meiosis. And the reason being is, remember when I said growth and reproduction were kind of connected in many ways. And they really are. And so one of the things that's really, really important is to then identify what the purpose of both of these things are and these processes. So you've heard of these processes, but what are they? So mitosis is related to growth, and its predominant job is to create identical, we call them daughter cells, right? So you want to go from one cell to two cells that look exactly the same. So here you have 100% of your DNA, here you have 100% of your DNA, 100% of your DNA. How do you get there? Well, you have to go through this middle zone, a middle process of the cell cycle. Oops, that was my eraser. I didn't want that. You have to go through this middle stage of the cell cycle where you actually have 200% of your DNA, right? Synthesis, split those apart so that then we can end up with two identical cells, exact copies of one another. But when we're talking about reproduction, let's draw a little image off here to the side. When we're talking about reproduction, we're not talking about making clones. We're not talking about making genetically identical people. We're talking about increasing genetic variation. We're talking about having a completely unique person. And so this process of meiosis is coupled with the idea that genetic variation is essential. We want as much, if you remember when we talked about this, we want as much variation as possible because then we're always ensuring that somebody can survive. So how do we get as much variation from possible, or as much variation from possible for an individual? Well, let's couple this with other things we know. If this is you, right? What are you made of in terms of your DNA? Well, you're made of your mom and you're made of your dad, and they each give you 50% of their DNA, and that makes the 100% of your DNA that's you, right? So if you are two sets of 23 chromosomes total, you're getting one set of 23 from your mom and you're getting one set of 23 from your dad. And that makes perfect sense, except for the fact that each of your dad's cells has 100% or 2 times 23rd of his DNA, and your mom, all of her cells have 100% of her DNA, or 2 times 2 sets of 23 of her DNA. So the question is, what process is it that's going to increase this genetic variation and get us from mom's 100%, get us exactly 50% of that, so that then when you combine mom's 50% and dad's 50%, now you're at 100%. If we did it a different way, if mom started 2 to the 23rd and dad started 2 to the, or not 2 to the 23rd, excuse me, 2 times 23, right? They have their 100% of their DNA, 100% of their DNA, and they give all of that to you. What are you looking at? Well, now you're looking at 4 sets of 23. You're looking at 200% of the amount of DNA that you would want. And guess what? The next generation, right? What's that? Oh, now all of a sudden you have 8 times 8 sets of chromosomes, so that's not good. So we need a way to divide or to get exactly half of the parental DNA. And that is the process of meiosis. So the purpose of meiosis is very much different from the purpose of mitosis. And the purpose of meiosis is to create unique gametes. And gametes are egg and sperm. So we want unique gametes that contain 1/2 of the parental DNA. And it's a very specific 1/2. It has to be exactly one set of the 23 chromosomes. You can't have just 23 random chromosomes. You need one number 1, one number 2, one number 3, etc., etc., etc. So the model below is essentially asking us, how do you think the meiosis, what do you think the meiosis looks like? What do you think that process looks like? Again, this is a thought experiment, not, it is not, I'm not looking for you to YouTube this. So if we go down here to this model, this is what you're looking at. This is your assignment. And so you have some guiding questions on the left, which hopefully I started to address. And then you have these models on the right. And so I'm going to start your model off with mitosis. And this is a starting cell. So this starting cell has not been duplicated. So this is like still in G1. Right? And then what we want in mitosis, we want identical cells. So at the end, what do I want to end with? I want to end with a cell that looks exactly the same. So I want a cell that looks, I want two cells, or excuse me, not one cell, I want 2 cells that look exactly the same as my starting cell. So the question is, what has to happen in order to get there? Well, if this is in G1 and I want to move this without drawing a whole nother one, I want to move this to prophase, right? I'm going to cross this out and I'm going to say prophase. Well, what will have happened in prophase? Well, they all would be duplicated. So I would have this, all of my chromosomes would be duplicated because they went through synthesis, right? When I draw this, I'm going to lose the size, but hopefully you can maintain the size here. I'll do my best. So now I'm in prophase, and now I'm going to model how I get to the ending result there. And so what I know is that in metaphase, right, if I were to draw out metaphase, all of the chromosomes are going to line up on this metaphase plate here. It doesn't matter what order they line up, right, because ultimately, we want one of each of these to end up on either pole. So what matters is not whether blue lines up next to blue, i.e. chromosome 1 lines up next to chromosome 1, or chromosome 17 lines up next to 17. What matters is that they're all oriented the same way. What you don't want is you don't want one chromosome to be oriented this way, right? Because then how is it supposed to get pulled apart? If that makes sense. So then I'm going to attach my spindle fibers, right? So spindle fibers are going to attach to the centromere. And so one side will attach there, the other side will attach there, so that we can pull each of these sisters apart, and so that we're going to end with one little light green, one little light, or one big light blue, one little dark green, one big dark blue, right, in each of the different poles of the cell, so that we can create 2 identical cells. So your task is now to complete the model for mitosis, and then to take a look at what the purpose of meiosis is and see what you end up with in terms of creating gametes that have 50% of the DNA, and on top of having 50% of the DNA, have genetic variation. Okay, so that's what we're looking for in the next model. I hope this was helpful. I hope you feel like you have the ability to give it a go. Again, don't use the internet, just give it a shot, and we'll talk about it in class. See you later.